Biology.
Yes, hello there and welcome. Now today let us look at genetics, and we are going to look at almost 100 questions of genetics just to revise the topic. So we begin with the first question. So the first question is asking, define the term genetics. So what is genetics? So simply genetics, this is the study of inheritance and variation. That is the definition of genetics. For example, we have been asked to define what is genetics. So for the question number 2, the question is asking, differentiate between continuous and discontinuous variation. So that is the question, we differentiate between continuous and discontinuous variation. So for this, we are also going to give different examples so that we try to understand even better. So for the continuous variation, we see that this continuous variation is a type of variation whereby there are a wide range of differences in character in a population. So the differences between the population and a specific character is very large. So as you can see, these few examples that you are giving, the examples of continuous variation. So we have height in organisms. So apart from height in organisms, we have the different skin colors, as you can see. So for the after the skin color, we can also have a variation of body size or body weight. After that, you have variation of intelligence, because it's not that easy for you to measure who is more intelligent than the other in terms of value. So it is not that easy. So apart from that, we have the feet size. Some people have a very small feet size, some have a very long a very large feet size, etc. and etc. So this continuous variation, this is whereby there are clearly defined differences between between a specific character in a population. So there are clear defined differences. Maybe for instance, some people can do somersault while some people cannot do somersault. So you see, there is a very wide difference between the characteristics. So in this discontinuous variation, now we tell the students it's either you have it or you don't have it. It's either you can do it or you can't do it. So that is for the discontinuous. So there is a very wide range of differences between a specific character in a population. So examples of discontinuous variation, as you can see, we have the different blood groups. Some people have blood group A, some people have blood group B, some people have blood group AB or blood group O. So that is discontinuous variation. So there is a real big gap between one character and the other. Apart from that, the other difference is gender. So you can see that we have some people who are males, some people who are females. So we have that very big gap. These people are males, these people are females, to bring that difference. Apart from that, so the other one you can see is tongue rolling. So the tongue rolling is not only that you can put your tongue to look like a letter U. So there are different ways of tongue rolling. So most people can do the letter U, but most people cannot do the rest of the tongue rolling. So that is what we mean in this case. So apart from that is the earlobe type. So there are some people, this earlobe is attached to the body, and there are some, this earlobe is just hanging freely. So that is also discontinuous variation. Apart from that, you have the eye color and many more. So we have different discontinuous variation. There are even over 500. So these are just examples. So remember, in this discontinuous variation, there is a very wide gap in characteristics that is being observed by it's being observed in a population. So it's either you have it or you don't. It's either you can do it or you can't do it. So let's go to the next question. So the next question is asking, letter B was asking we differentiate between homozygosity and heterozygosity. So homozygosity and heterozygosity. So remember, this one mostly we deal with the alleles that are found in the gene. So this one mostly deals with the alleles. So what is this homozygosity? So homozygosity simply occurs when both copies of the gene are similar. So like when both of the gene copies, that is the alleles. So if both of the alleles are similar, that condition is called homozygosity. If you can look at the examples what you are giving, we have given examples. We have, for example, we have AA. Those are the alleles. We have allele A and A. We have allele B and B. We have recessive C. We have recessive D, So if the alleles are same, this condition is called homozygosity. But what about heterozygosity? So for the heterozygosity, remember, this is now when the alleles are different, or gene controlling a characteristic are different. So if the specific gene which controls the characteristic are different, this condition therefore is referred to as heterozygosity. So that is that phenomenon or condition. It is what is being referred to as now heterozygosity. So for the next question is asking, we differentiate between allele and trait. So what's the difference between allele and trait? Because mostly in some books, or rather, some people call it trait, some other people call it characteristic. So if you want to call it characteristic, good for you. If you want to call it trait, it is also good for you. So simply, a trait is that physical outlook you observe in an organism, or simply you can say a trait is a characteristic that is observed in an organism. So that is the trait. Like for instance, you can see my skin color, I am an African, so my trait is dark. For instance, you can see maybe Europeans, their skin color is white, so their trait is white in color. So maybe the hair size, maybe the eye color, just anything you can see in an organism is called a trait or is called a character, even including the height, including the body size. So just anything that you can see in an organism, that is what is called a trait. And what about an allele? So simply an allele is a different version of a similar gene located on the gene locus. So that is it. So that is the allele. For example, you can see like what you are doing, we are saying that for the homozygosity, we said we have capital A, capital A. So that gene has two alleles. So the first allele is capital A, the second allele is capital A. Maybe if you can try to look at the heterozygosity, we can see we have a capital A and we have a small a. So these are two alleles. We have the dominant A, which is capital. We have the recessive A, which is now the small letter. So we have mentioned gene locus. So what is a gene locus? So specifically, gene locus means now that specific location where a gene is located on the chromosome. That is what we refer to as the gene locus. That specific location in a chromosome where a gene is located. So that is what is referred to as a gene locus. So don't forget that. I think this question you're going to see it in the upcoming question, so it will be good to go. So question letter D is asking, we differentiate between dominant and recessive allele. So we have already said this, but let's just differentiate. So letter D is asking, we differentiate between a dominant and a recessive allele. So like, what is a dominant allele? What is a recessive allele? So simply we see that a dominant allele expresses itself both in homozygous and in heterozygous state. That is a dominant allele. It can express itself both in homozygous state, that is on a cross or a Punnett square. It can express itself both in homozygous state, or it can express itself in heterozygous state. That is a dominant allele. I've given examples as that capital A, and then also we have capital B. So as long as you see any capital letter to be represented in a cross, always know that that is a dominant trait. So for the recessive trait are always represented by a small letter. But then the next one is asking, we define what is a recessive trait. So a recessive trait, we see this is a trait which only represents itself in homozygous state and not in heterozygous state. So it only represents itself in homozygous state and not in heterozygous state. So the question was asking, we differentiate between a dominant allele and a recessive allele. So a dominant allele, we have said that this is an allele whereby it represents itself both in homozygous state and in heterozygous state. While a recessive allele, we have said that this is an allele that only represents itself in homozygous state and not in heterozygous state. So that is that. So the next question is asking, what is a gene locus? So that is the next question. It's like the previous one that you had seen before. So this question is asking, what is a gene locus? So if you have been asked what is gene locus, what are you going to say? So gene locus, as you can see in this diagram, I've tried to highlight. So a gene locus, remember, this is the specific location whereby a gene can be located on a chromosome. That is now the definition of a gene locus. So if you're talking about many gene locus, you're going to call it gene loci. Some people will call it gene loci. So gene loci is plural, while gene locus, that is supposed now to be in singular formation. So that is what we call a gene locus. So the specific location whereby a gene can be located in a chromosome is called a gene locus. And each gene locus controls a specific characteristic in an organism. So you can have maybe the locus, that specific part of the chromosome, that is the locus controlling the eye color. You can have a specific part of the locus controlling maybe the hair color, maybe the height, maybe also different aspect, different traits or different characteristic for that offspring that offspring is supposed to have. So that is what we call a gene locus. So the next question is about laws of heredity or law of segregation. It just simply means the same thing. Laws of heredity, segregation. So more or less, it's just a similar thing. So I've been asked, state the first law of genetics. So, like, what is the first law of genetics as per Mendel's experiment? You remember? He took the garden pea plants, he crossed the short ones, then he crossed the tall ones, then he crossed a crossbreed in order to get offsprings, and then finally come to a conclusion. So state the first law of genetics. So the first law of genetics states that a characteristic of an organism is basically determined by the hereditary factors that occurs in pairs which are called the alleles. So if the parents were tall, maybe let's say the dad has a dominant tall gene and the mom has a recessive short gene. So the offspring is going to obtain the dominant characteristic, only one characteristic from the height. They are either going to obtain the tall character or the short character. That is what it says. So the characteristic of an organism is determined by the hereditary factors occurring in pairs called the alleles. So the offspring is either going to obtain the tallness of the dad or the shortness of the mom. So that is the first law of heredity. Simply, an offspring only attains, or an offspring only obtains one of the traits from the parent. So, like for Mendel, he didn't base his fact that the offspring can be able to obtain 50-50 from the parent. So the first law of heredity only tells us that an offspring obtains one characteristic from the parent. So you can see that it is only one of these factors that can be represented in a single gamete. So it is only one character to be represented in a single gamete, which is also going to be represented in how the organism that is going to be formed, as you can also see, being stressed by the law of segregation. So the next question is asking, state the importance of genetics. So what are the importance of genetics? Why should we study genetics? Or in short, why are we studying genetics even right now? So what are the importance of studying genetics? So the first importance of studying genetics is that we see it provides us with knowledge of plant breeding to obtain good and high yields of crops. So through genetics, so we can be able to breed plants. We can be able maybe to use the different chemicals on plants, like auxins we studied in Form 3, like cytokinin, like florigen, in order to obtain very high yields of the plants of crops. So through genetics, you can see that we can be able to modify genes to obtain fast-growing organism. Maybe, for example, the chicken that are being sold around, 3-week-old chicken. So in 3 weeks old, it was a chick, but now it has grown, it is large, it is mature and ready to be consumed. Also in the greenhouse, we see that plants are being planted. So a normal maize should take 3 months, but now this maize in the greenhouse only takes some few months, maybe 1 month. Then the maize is mature and it's ripe, so it can be fed on. So also through genetics, we can see that we get knowledge of breeding, which leads to disease-resistant animals. So through breeding, through crossbreeding, so we can be able to obtain animals that are disease-resistant. So also through genetics, we see that we can get knowledge of genetic counseling and also be able to provide good knowledge for childbirth or good knowledge for the parents who are unable to obtain children. So through a genetic counselor, the genetic counselor might sit down the parent, the male and the female, or the mom and the dad, and advise them concerning their genes. It is through their genes that maybe they have obtained this offspring. So maybe if the parents are fighting that, if you look at them, none of them is an albino, but they have obtained a child who is an albino. So the genetic counselor may come in, may study the backgrounds of both of these parents, and then advise them accordingly. That maybe your great-great-grandpa had this trait of albino. So, like you, the dad, or you, the mom, this trait has been masked all through the years, but now you getting the child, you have not gotten an albino. So it is not abnormal, these genes are from you, blah, 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 things like that. So that is not the genetic counselor who can now counsel people based on their genes and what kind of children they are going to get, or what kind of children they are expecting to get in the future. Also through genetics, we see that, so maybe if someone has touched your thing or someone has stolen anything, so the forensic scientists, they can come and get the fingerprints of everything that was touched, and then eventually arrive to a conclusion of who stole the item based on the fingerprints. So through this genetics, we see that different legal disputes can be settled, especially through fingerprinting, because that is the most important. Also through rape cases maybe. So the forensic scientists, they can come maybe to the victim who has been raped. They collect the fluids of maybe the person who did it. They collect the fluids. They take it to the lab. They match the DNA, and then they get the culprit who raped the victim. Therefore, if you have been raped, so make sure you don't shower. Just go first of all to the hospital. So let them conduct the experiments. After conducting the experiments, then now you can go and shower. Because if you shower, you're going to wash away all the evidence. And if you wash away all the evidence, it will be difficult for the legal people to get the actual perpetrator of the misconduct. So also through genetic engineering, unwanted offsprings can be prevented. Maybe for instance, in this family, so we have people who are dunderheads in this family. But in this family now, so we want an offspring who is intelligent, an offspring who grasps everything very fast. Through genetic counseling, through genetic engineering, this defective gene of not understanding anything, of lack of intelligence, so it can be removed and then replaced by a gene which is for intelligence. And then after that, the DNA is wrapped together again and then taken to the gamete, and then this gamete fertilized, and then the offspring to be obtained is going to be super intelligent. So, and that is how most of these big countries, they make geniuses. So unasikia mtu ni genius. So that is what most of them do. So they go and take that non-intelligent gene, they remove it from the chromosome, then they put now the gene that is for intelligent. So if that child will be born, that child will be super intelligent. So also, so you can do that for the hair color. If you want the child you're going to get to have very long hair, you can do genetic engineering. If you want them maybe to be this tall, this short, if you want them, so in short, through genetic engineering, so you can be able to manufacture the offspring or the baby that you want. If you want your child to look like this, all the fantasies that you think of, if you want your child to look like that, so you can just go to hospital, I guess Karen Hospital and Aga Khan also do the procedure. So you can just go to those hospitals, you give them maybe their sperm, your ovum, they remove those genes that you don't want, they put everything that as per your specification, then they create that offspring for you. And I guess it's quite expensive. So also through Human Genome Project, that is still the importance, through Human Genome Project, so different conditions, health conditions have been treated, like for example, heart attack and stroke. So treatment for those conditions have been obtained, whereby, so through this genetic engineering, so those conditions that lead to this heart attack and stroke have been dealt with accordingly. So we go to the next question, as you can see, apart from genetic engineering. So the next question is talking about a pedigree chart. So it's saying, use H in the pedigree chart below for the dominant gene where both parents are phenotypically normal. So I've been told that both parents are phenotypically normal. But as you can see in the pedigree chart, you can see that one offspring, we have one hemophiliac male. So the key says that the circles represent the females and then the boxes represent the males. So a blank box represents a normal male, while a yellow box represents a hemophiliac male. So I've been told, what are the parental genotypes? What are the parental genotypes? So I've been asked, use H in the pedigree chart. Use H. So the condition we're being told, this condition is hemophilia. Hemophilia is a sex-linked gene. So sex-linked gene simply means a gene with a characteristic or a trait which is transmitted together with the sex genes. So the sex genes, remember, we have two of them. We have the X and the Y. The male is XY, while the female is X and X. So those are the sex genes. So this hemophilia is now a sex-linked gene. It means that as this gene is being transmitted, it is transmitted together with the sex genes. So as the gene for determining gender zinaingia to make the offspring, so also this gene for hemophilia, it's like it is bound to that X and Y. It enters together with it. That's why it's called a sex-linked gene. Now, for all the sex-linked gene, for you to represent them, you must represent them first of all using the gender genes, which is X and Y, and then you put that gene now on top. That is to show that this is a sex-linked gene. So you don't just say letter H and small h. That is wrong. Since it is sex-linked gene, so you must first of all include the genes for the gender, which is X and Y, for males X and X. And then now you put that gene on top of the H allele. You must put it on top of the H allele and not the Y allele, because it is the H allele that carries this sex-linked gene and not the Y chromosome. So the question is asking, what are the parental genotypes? So for the parental genotypes, we see that for the male we see that the male is normal. And then we have been told, use H to represent the dominant gene. So capital H represents that who is normal. A small h will represent that who is not normal, who has hemophilia. So remember, capital H represents normal. Small h represents the recessive, who has now the hemophilia. Now in this case, we have been told, what are the parental genotypes? So for the male parent, we see that they are normal. So for their genes, that is why their genes remain X, capital H and Y, because the male is normal. So for the female, we see that also the female is normal. So for the female gene, we see it remains to be X, capital H, and then X, and then small h, to mean that the female was a carrier. Why is it that the female is a carrier? It's because, remember, we have an offspring that is hemophiliac. Males cannot be carriers because males only have one X chromosome. So there is nowhere in this world you will ever see that a male is carrier of hemophiliac. It can never happen. Why? So if you have been asked in exam, you'll say it's because males have one X chromosome. That's why they can never be carriers. So it's either males have that disease or they don't have the disease. That is it. So, for example, so if the male here was affected, it could have been small h. The male could have been hemophiliac. But you have been told that the male is normal. So it means, and the males have one X chromosome. So it means that the gene for the male is X, capital H, then Y. Females are carriers. Why? Because females have two chromosomes. So, like if they have two alleles. So if one allele is affected, the other allele is normal. So this allele that is normal always dominates over the one that is recessive. So that's why the female is a carrier. Like phenotypically, she is normal. Phenotypically, you can see the female is normal. But now the traits inside the gene, she has that characteristic of developing an offspring who is hemophilia. That is what we mean by saying a carrier. Therefore, for the females, the genotypes of the female is X capital H and X small h, to represent that the female is hemophiliac. So part B of the question is asking, state the genotypes of the offspring. So what are the genotypes of the offspring? So the first offspring, we see that they are normal males. They are normal female. The next offspring, we can see they are normal males. The third one, we can see normal female. But the fourth offspring, we can see that is hemophiliac male. So if The fourth one is hemophiliac male. It means that it's X, small h, and Y. So remember, we don't have male carriers. It's either the male has the disease or is normal. Therefore, in this case, we have X, small h, and Y. That is why we have now this male who is hemophiliac. Now that summarizes that part. So we go to the next question. So I guess you're going to find more of these questions of pedigree chart. You're going to revise it even more with the upcoming questions. So we had question, we have question C. We continue. We have question C. We have been told that if a woman carrier marries a normal man. So I've been told that a woman carrier. So remember, woman carrier, we have X capital H, X small h. So we have been told that if a woman carrier marries a normal male, show the genotypes and the phenotypes of F1 generation. So remember, capital H means a normal dominant gene. Small h is a recessive, which now means that this person should be having the hemophilia disorder. As you can see, this genetic cross, the genetic cross, try to summarize now the genetic cross between now the normal male and the hemophiliac and the carrier female. So that is it. Normal male and the carrier female. That is exactly what we have been asked in the pedigree chart. We have a normal male and a carrier female now getting the offspring. So as you can see, the offsprings down there in F1 generation, we have a normal female because it is X capital H, X capital H, purely normal female. Then the next one, we have a carrier female. Why a carrier female? Because the first allele is capital H, which symbolizes normal, but there's the second one is X small h, which symbolizes now she is a carrier. She's normal phenotypically, we can see she's normal, but in her blood, in her genes, so she has the genes of providing an offspring with hemophiliac. Then the third one, we see that we have a purely normal male. Then the next one, we have a hemophiliac male. So remember, males can never be carriers because males have only one X chromosome. It's either they have the disorder or they are normal. So always remember that. So males can never be carriers. Then remember that reason. Now we go to the next question. It's asking, explain two applications of genetic engineering. So what are the importance? Application, it's like saying importance. What are the importance of genetic engineering? So genetic engineering simply means it's now taking that DNA, we manipulate that DNA, we remove the genes that we don't want, then we put the genes that are important, and then we zip the DNA. After zipping the DNA, we take it back to the chromosome. After taking it back to the chromosome, we take it to the gamete, we fertilize the gamete. Then we have the offspring that we have manufactured, the offspring that we want, with all the characteristics that I want, is now the offspring that is going to be formed. Genetic engineering, so that is applicable in medicine. Genetic engineering can be used in very many avenues in life, not only in childbirth and medicine, in very many avenues. So simply remember that genetic engineering is the modification of an organism DNA by using scientific methods. So if we alter the DNA of organisms using scientific methods, that is called genetic engineering. So we are going to obtain the characteristics and the traits that we ourselves want. So the first application of genetic engineering is that we can use it to make insulin, we can use it to make vaccines. Also, you can use it also to make different growth hormones. Like for example, you're going to see for these people having this condition called achondroplasia. So they are mature people, but they are very short, always very short. Anyway, I think we are going to get that question. We are going to look at it. Also, through genetic engineering, you can be able to get crops that are resistant to drought. Yeah, so you can be able to manufacture crops that are resistant to drought. For example, we have this breed of maize which is called, it was called Katumani. So this Katumani is a maize breed that can be able to grow in arid and semi-arid areas, and it will still grow, it will still produce maize, very healthy maize. So through that, it is through genetic engineering. So also through genetic engineering, you can be able to produce animals that are resistant to different diseases. So different animals resistant to different diseases, through genetic engineering, we can be able to obtain them. Apart from that, you can also be able to obtain faster growing animals. You remember in the previous question we said you can be able to obtain chicken that are 3 weeks old. So yeah, 3 weeks old chicken, they were born 3 weeks ago, they were hatched 3 weeks ago, but now they are mature and they can be eaten. So that is through genetic engineering, through providing with the different growth hormones they are injected with, then they grow very fast and then they are ready for the market. Also for the maize, the normal maize should take 3 months, but the maize that have been manipulated genetically, they can even grow for 2 weeks. So I've planted it today, after 2 weeks, after 1 month, the maize is grown very tall and it's ready to be harvested. So also by that you can be able to use genetic engineering to develop growth hormones, different growth hormones in the body. So in order to enhance growth, especially in humans, so not only plants and also in humans. As you can see, these individuals, you can see these individuals are mature. You can also see their faces, you can see their bodies. They are very mature. But now the problem is they are now, the height wise, they are short. They are very short. Maybe their height can even be about 130 centimeters. So if you can be able to see 130 centimeters is quite short. So my height specifically, I am 180 centimeters. So being this, the 180 centimeter, so I can assume 130 centimeter should reach me somewhere here. So they can be about 130 centimeter or even shorter. So remember, height, we talked about it, it's continuous variation. So for this continuous variation, some can be maybe here, some can reach me here, some can reach me here. So it doesn't have that clear kick where it's supposed to be. So it's a continuous variation. So they can be short. So this condition is called achondroplasia, as you can see. So it's called achondroplasia dwarfism. So and then these people having achondroplasia, also their faces somewhat resemble. They look, all of them, they look like brothers, all of them, they look like sisters. So we are going to see in genetics that it is mostly protein number 26. It is gene number 26 to control the protein distribution in the body, which has been affected, among others. So this condition, remember, it's called achondroplasia. They are not sick, but it's just a condition. So it's a condition which leads to dwarfism. So that is it. So the next number is asking, differentiate between gene mutation and chromosomal mutation. So what is gene mutation? What is chromosomal mutation? Just from the word gene. So if we change the structure of the gene, therefore that gives us now the gene mutation. So if we change the structure or the sequence of the DNA in the gene, that simply is gene mutation. So remember, for the DNA, we said it has the different bases. We said we have the adenine, thymine, guanine, cytosine. So if we change any of this sequence, so the default should be like this. So if we change the default sequence to something different, that is what you are calling gene mutation. If we change the sequence of the DNA, that is what we call gene mutation. As you can look at these few diagrams that we have here, we have the different nucleotides. You can see the first nucleotide has been mentioned, and then you can see the other diagram, normal gene, then a mutated gene. So if there is anything that has affected the structure of the gene or the sequence of the DNA, that now gives us a gene mutation. While chromosomal mutation, this is the change in the structure and the number of chromosomes in a cell. So if you change the structure or the number of chromosomes in a cell, that gives us chromosomal mutation. So I think we're going to look at this mutation in there's a question to deal with this mutation. But now we have been asked the definition. So if you change the sequence of the DNA, that gives us gene mutation. If we change the structure of and the number of chromosomes, that gives us chromosomal mutation. A good example of chromosomal mutation is non-disjunction. So in non-disjunction, so whereby some of the cells we are going to have, so the normal number of chromosomes in a cell should be 46. So now, in this non-disjunction, it's a chromosome mutation. In this non-disjunction, some cells may have 45, some cells may have 47, etc. So you see, if we change the structure or the number of chromosomes in a cell. That now gives us now the chromosomal mutation. These are the mutations that we are going to talk about. So we're going to talk about mutation, which are duplication. We're going to talk about deletion. We're going to talk about inversion. We're going to talk about substitution, translocation. So as you can see them, these ones you're going to discuss them. There's a question to deal now with these things specifically. We're going to deal with them, talk them, discuss them, etc. So the next question is saying that a patch of white hair on the head is a dominant condition in humans. Then a woman with a white patch of hair married a normal man and they had a daughter with white patch. So remember, so in these conditions we are being told that now the thing that happened is that so the white patch on the head is now the dominant condition. So being normal in that family now is a recessive condition. So being told that a woman with a white patch married a normal man. So after marrying the normal man, we see that the daughter they obtained, had a white patch on the head. So remember, having a white patch is not the dominant condition in that family. So the daughter married a normal man. So the daughter who had the white patch married a normal man, as you can see in the question. Then state the likelihood that the first child will have a white patch. So we have been told to use letter P to now represent the dominant gene. So the dominant gene, remember, is the gene for having the white patch. So use letter P now to represent the dominant gene in this case. So as you can see, this is the genetic cross for this question. But let's now explain this genetic cross so that we see. So the answer should be there is 50% chance likelihood that they are going to obtain a daughter who has a white patch. So the chance is 50%. Let's now discuss this genetic cross that we have here and see. So remember, so for the woman, we have been told that the woman has a white patch on the head. So they have a white patch on the head. So this means that the woman is a carrier for that condition. So she has a carrier for being normal, but she has also now that condition of having a white patch. So that's why you can see the genotype of the woman is represented as heterozygous, capital P and small p. So for the male, remember, for the male, you have been told that the male is normal. So if the male is normal, remember the gene for being normal is small p, small p. So according to this family now, being normal is recessive. Having a white patch, that is now the dominant condition. So after that now, you follow up with the gametes. After gametes, you now show the F1 offsprings after showing the genetic cross. So as you can look at the offsprings, so we have the first offspring, we have the second offspring who has a permanent white patch, the third offspring is a carrier, and the fourth offspring to obtain, the fourth offspring should be normal. So the first offspring should be a carrier, the second offspring should now have the permanent white patch on the head, not a carrier, and the third one should also be a carrier. So what is the probability of the first child to have a white patch of hair? So that white patch of hair, therefore, the probability that they're going to have a white patch is 50%, as you can see. So for the white patch, that is what is being represented by the capital P and then the small p, as you can see it. So let's go to the next question, which is asking state 4 main differences between now the DNA and the RNA. So what are the differences between now DNA and the RNA? So the first difference is that we see that we have drawn this table, so we're just going to go through this table as it is. So for the DNA, we see that the DNA is double-stranded, while the RNA is single-stranded. So the DNA is double-stranded, it's on both sides, having now the nucleotides, and then the RNA is single-stranded. So RNA copies the information on one side of DNA, then takes it to the ribosome for protein synthesis to take place. Remember that. So the other difference in DNA, you can see it has a deoxyribose sugar, while the RNA has a ribose sugar. Now, since the DNA has now this deoxyribose sugar, that's why it's called deoxyribonucleic acid, because of that deoxyribose sugar. The RNA is called RNA because of it has a ribose sugar. That's why it's called an RNA. So remember, DNA has a deoxyribose sugar, that's why it's called a DNA. RNA has a ribose sugar, that's why it's called an RNA. So don't forget that. So the other difference is that the DNA, we see that it has the base thymine, it has the nucleotide which is thymine, it has a base thymine, while the RNA has the uracil. So anytime in the DNA or anytime in these structures or in the helical structures, you're going to see that we have adenine, we have thymine. So always know that that should be DNA. Anytime you will see in these helical structures, we're having uracil. Always note that that means that that structure is an RNA. So for the DNA, the adenine combines with the thymine. But for the RNA, the adenine combines with the uracil. That is how you differentiate between the DNA and the RNA. The DNA, remember, it has the thymine. The RNA, remember, it has the base uracil. So the DNA is found in the nucleus and the mitochondrion of the cell, while the RNA, yes, is found in the nucleus. It is also found in the cytoplasm and also found in the ribosome. So why is the RNA found in nucleus, cytoplasm, and the ribosome? So remember, the RNA is a single strand. It goes to the DNA. It's like a messenger. We have a messenger RNA. So it goes to the DNA. It copies that structure or one side of the helical structure. After copying it, that is in the nucleus. Everything is happening in the nucleus. After copying it in the nucleus, it goes to the ribosome. But how does it reach the ribosome? It passes through the cytoplasm. So from the nucleus, after copying, it enters the cytoplasm, it moves through the cytoplasm and then now to the ribosome. That's what you're saying. The location that by which that you can find an RNA is in the nucleus as it copies, is also through the cytoplasm as it moves to the ribosome, and finally to the ribosome, where now protein synthesis is going to take place. So those are the locations of RNA. So remember, for the DNA, we find it in the nucleus and also in the mitochondrion, to denote the amount of energy to be produced for a specific reaction. So also, apart from that, we see that the DNA contains genetic materials, the genetic materials of that offspring. So the DNA contains genetic materials, while the RNA now carries this genetic material. It now carries the genetic materials from the DNA or from the nucleus and to the ribosome. So remember, the DNA now contains that helical structure of DNA. It contains now that genetic information, the specific type and amount of protein to be produced. So after now the DNA having this information, now the RNA, it comes and copies this information of the DNA. It takes this information to the ribosome for the ribosome to make that protein. So that is exactly how these two things relate. So the DNA codes the information. This is the protein that we need. The RNA, it copies this information and takes it to the ribosome, so that now the ribosome will make that specific protein that the DNA has coded for. So we go to the next question. As you can see, it is asking about sickle cell anemia. So the question is saying, sickle cell anemia is a hereditary disease caused by a mutant gene which changes production of normal hemoglobin HbA to abnormal hemoglobin HbS. So sickle cell anemia is a hereditary disease. So hereditary means it can be able to be transferred from the parent to the offsprings. So if this lineage, they have sickle cell, chances are very high they are going to give rise to offsprings who have that same condition. Unless maybe there's some genetic crossbreeding that happens so that they do away with this gene completely in the lineage. But if it's continuous like that, and if more so the sickle cell is a dominant gene, so it's going to happen that now sickle cell will be recycled in this family over and over again. So sickle cell anemia is a hereditary disease caused by a mutant gene which changes production of normal hemoglobin HbA to abnormal hemoglobin HbS. So question is asking that what are the possible phenotypes of the heterozygous man? what are the possible phenotypes of a heterozygous man and heterozygous woman for this trait. So the heterozygous man, so remember heterozygous means that they are normal, they have the normal gene, but they have also the sickle cell gene. So the normal gene is now the dominant gene over the sickle cell. So a normal man is HbA to mean that they are normal, and HbS to represent that inside their gene there is this sickle cell trait. And also for the woman the same. A normal woman is HbA to represent that phenotypically they are normal, but now they have this gene which can be able to transmit sickle cell trait to an offspring. That's why, as you can see at this, if you can look at the genetic cross, we have said that we have HbA, HbS. These people are normal. If you look at them, they are normal. The sickle cell trait is not affecting them. They are normal. But if they get an offspring, they can get an offspring who has this disorder or who has this disease. So we have been told we show a cross between a normal sickle cell man and a normal sickle cell carrier female. So as you can see, this genetic cross, we have crossed the genotypes, which is HbA, HbS, crossed with HbA, HbS for the female. So the offsprings you are going to obtain, the first offspring is going to be purely normal. You can see HbA, HbA. This offspring will be purely normal. So the second offspring you are going to obtain is an offspring who is a carrier for the sickle cell. That is HbA. They are physically normal, but in their genes, they have this trait of getting an offspring who is sickle-celled. So the next offspring also is HbA, HbS to mean that physically they are normal, but in their genes, they have the trait of getting an offspring who is sickle-cell. Then finally, we have HbS, HbS. So the fourth chance of getting an offspring, we can get an offspring that is purely sickle-celled. So physically they are sickle-celled, and also getting an offspring, they'll raise offsprings that are also sickle-celled. So remember in heterozygous, remember the heterozygous we meant? So heterozygous meant that the alleles are different. Alleles are not the same. So remember, homozygous means that the alleles are similar, but heterozygous means that the alleles are different. So remember that heterozygous means the alleles are similar. Heterozygous means that the alleles are different. That's why we have HbA, HbS. The alleles are different. So maybe you could have said, so you can see the fourth chance. The fourth chance of getting an offspring, that is HbS, HbS. So that is homozygous sickle cell because the alleles are similar. You can also see the first chance is HbA, HbA. That is homozygous state whereby the alleles are similar. But now the second and the third chance, the offsprings are they are normal, but they have the trait of sickle cell. So this condition HbA, HbS, that is now what we call heterozygous, if you can be able to remember that. So that's not something to forget. So remember, heterozygous, alleles are different. Homozygous state, alleles are similar. Part B of the question is asking, what proportion of the offspring will be severely anemic? Severely anemic, anemia, sickle cell. That's what it's called, sickle cell anemia. So severely anemic means that, it means that the offspring, if you look at the offspring is has sickle cell. You can see all the symptoms and condition, and also they have now the genes for producing an offspring who is also sickle cell. So that is what it means, severely anemic. If you can look at this, the cross again. So the offspring who is severely anemic is the fourth chance, HbS, HbS. So this simply will mean that, to answer the question, what proportion of the offspring will be severely anemic? So it is only 25%. 25% chance. So remember, these things down here, the offspring is not that you're going to, the first offspring to obtain will be normal, the second offspring to obtain will be having some sickle cell trait, the third one, then the fourth one. No. These are chances. So in all these are 25, 25, 25, 25. There's 25% chance of obtaining an offspring who's normal. There's 50% chance because it's both of them, HbA, HbS, HbA, HbS. There's 50% chance of obtaining offsprings that have some condition of sickle cell in them. Some of the white red blood cells are normal. Some of them, they have some sickle cell trait. Then now the last one, HBS, that is 25%. So these things are just chances. 25% chance of getting a purely normal, 50% chance of getting some sickle cell trait, then 25% chance of getting now severely anemic. So the answer there was 25%, since in all these 4 chances, it is only 1 chance that we can get a severely anemic child. So mildly anemic, so mildly means that they have some trait of sickle cell. This is the HbA, HbS, HbA, HbS. So mildly anemic, so we have 50% chance. Because remember, for the 4 chances, it is only 2 chances that you can get mildly anemic individuals. So it is 50% chance. So we have mentioned anemia. What are the signs and symptoms of anemia? I have tried to get a diagram to explain. As you can see this figure here. So someone having anemia, they are going to be fatigued. They are constantly to feel tired. So after feeling tired, they are going to feel dizzy and faint. The heartbeat rate has also increased with increased in mood. They are going to feel chest pain because the heart is constantly trying to pump the few red blood cells in the body to supply the body organs with enough blood. Eventually, if it's not maintained, it may lead to heart attack. So if you can look at the eyes may have some sensation of being yellow. Why being yellow? Because these people have jaundice-like condition. What is jaundice? Jaundice is a condition whereby there is more plasma in the blood as compared to the red blood cells and the white blood cells. Plasma is yellow in color. That explains why most of the eyes of these people, it's going to be observed that it's yellow in color, to mean that there's a lot of plasma in their blood. After that, their skin is also going to be cold. Their skin is also going to be yellow to represent also jaundice-like symptoms. There's going to be intestinal blockage. So if you suspect any asthma conditions, or if you have anyone who is having sickle cell trait, make sure that they eat a balanced diet in order to always replenish the amount of blood in their body. And they are going to be okay. They are going to be treated. So you go to the next question, is saying complete the following diagram of the DNA by answering X, Y, and Z. So you complete the following diagram of the DNA by answering X, Y, and Z. So if you can look at this diagram, so it's a diagram to show now the unzipped DNA. So that is now the unzipped DNA. Let's see. So first of all, before we answer the question, what is the full meaning of the word DNA? So the full meaning of the word DNA, remember, it is deoxyribonucleic acid. That is the full definition of DNA. Deoxyribonucleic acid. That is the definition, don't forget. So according to this diagram, we should know first of all that in the DNA, the nucleotides, the adenine combines with thymine, the guanine combines with cytosine, and vice versa. So here, anywhere we are going to see guanine, we are going to put cytosine. Anywhere we are going to see thymine, we are going to put adenine. And anywhere we are going to see guanine, also cytosine, and vice versa. But now for this question, we have part X, Y, and Z, as you can see. So we have part X, we have part Y, and finally we have part Z. So part X, we have guanine. So guanine combines with what? Cytosine. So part X, you say it is cytosine. Part Y, we see Y should combine with thymine. So that means that part Y should be adenine, because adenine combines with thymine. Then part Z, we have C, which is cytosine. So cytosine, remember, it combines with what? Cytosine combines with the adenine, with the guanine. So if you do that, you are good to go. So question next is asking, we are being told that the color blindness in humans is caused by recessive sex-linked gene on the X chromosome. So we had discussed why is it called a sex-linked gene, because it is a gene, when transmitted, it is transmitted together with the sex chromosomes. Then we are being told that a carrier woman married a colorblind man. So remember the carrier term we said? So carrier means that phenotypically they are normal, but in their genes to get an offspring, they have this gene of getting an offspring who has that condition. So physically, the woman is normal, but if they get an offspring, they can get an offspring who has that condition. That is what we meant by a carrier. So I've been told that a carrier woman marries a colorblind man. So this colorblind man means they have that condition of colorblindness. So the colorblindness is predominantly their condition. So I've been told use capital C. It is capital. So make sure you get the term correctly. So remember, the question is not just saying C, but it's saying capital C. So use capital C to represent the dominant gene. So it means that if capital C represents the dominant gene, it will mean that small c is going to represent what? Small c is going to represent a recessive gene. So now, the first question is asking, what is the probability that a first child would be colorblind? So what is the probability that the first child will be colorblind? But before even we answer this question, like what is this condition which is called colorblindness? What is colorblindness? So simply this colorblindness, it's an ability of someone to be able to distinguish colors, especially green and red colors. This thing that people say that male are colorblind is not predominantly true, just that males cannot be able to differentiate the different shade of colors. So males are good in differentiating the primary colors, red, green, and red, yellow, and blue. But now the middle colors between red now and blue, those middle colors now is the one which becomes technical for them to be able to differentiate between them. So we're going to see females have many cons to differentiate colors compared to males. That is true anyway. Females have many cons. In the topic of reception, response coordination, we're going to go through this and see. So females have more cones. So remember the two cells to perceive vision, we have cones and the rods. These ones we looked at them in the retina of the eye, as you can see. So this topic we're going to discuss it in the topic of reception, response coordination. So females have more cones than males, therefore they can be able to distinguish between many colors as compared to males. So that is it. So maybe for me, I can see pink, I can see purple. Then if you ask me, I'll tell you that they are similar. This is pink, but a female will tell me, no, this is purple, or this is a shade of pink. So you see, they have many cons to differentiate them as compared to we, the males. So mostly, this condition which is now colorblind, remember colorblind is a condition which someone is unable to differentiate between colors, especially red and green colors, or the shades of red and green colors. So how does it come about? So simply this colorblindness mainly comes about when some cones in the retina, remember the retina, the cones? Some cones in the retina are missing or are defective. Do you people believe there are some people out here who don't even see red? If they look at red, they see red as gray. Completely they are colorblind. Hiyo sana ile imefika pabaya kabisa. Completely they are colorblind. If they look at blue, they see it as dark black. Not dark black, dark gray. If they look at red, they see it maybe brownish, coming to brownish, or maybe somewhat grayish. So like what exactly leads to this condition? So this condition is brought about when the cones of the retina, remember the cones are the ones which are enabling us also to perceive light, to see this light, bright light. They are the ones which receive this light, they make sense of this light after taking the signal to the brain, and then the brain interprets, and then I'm looking at color white, I'm looking at color red. So this color blindness condition happens when some cones in the eyes have been destroyed, or the cones are not functioning, or the cones are missing. Like now what we are discussing right now. A male who's colorblind, they marry a female who's colorblind. So they are going to give birth to children who have either missing cones or the cones are undeveloped. That's what brings this condition of color blindness, as we have said. So the cones of the retina, they have an enzyme which is called iodopsin. So this iodopsin is broken down. After being broken down, therefore we are able to perceive light of high intensity. So the cones, remember, they are used to see mostly when there is light. So when there is bright light, our eyes are using the cones to see. So how does it happen? So we are going to discuss it, but just a heads up. So the cones have this enzyme which is called rhodopsin. So this rhodopsin is always and constantly being broken down in the presence of light, so that now sense can be made out of what? The light which is entering the eye. So also the cones, apart from perceiving light of high intensity, cones can also be able to differentiate colors. Those are the two things you're going to study in the topic of reception. So they are used to perceive light of high intensity, and also they are used also to differentiate between the different colors that we have. Now, apart from that, apart from cones, we have the rods. So as you can see the diagram, we have the cones and the rods. This diagram, you can get it in KLB. So for the rods, on the other hand, the rods now are used to perceive light of low intensity. Because if you have ever been careful, so maybe when there's some darkness, maybe there's some dim light somewhere, it is difficult for you to note colors. By the way, did you realize it is difficult for you to see colors in the dark? Not really in the dark. Maybe like, for instance, there's some dim light. Or maybe it's at night, and then maybe someone is approaching, or a vehicle is approaching from very far. It is difficult for you to see the colors that are around. Or maybe in that dimly lit room, it is difficult for you to differentiate colors. Why? Because if there is minimal light, the cones which are used to perceive colors are not functioning. Therefore, the thing that is functioning in this case now is the other cells which are called the rods. So the rods are used to perceive light of low intensity, and also the rods cannot differentiate colors. So remember, the two functions of the cones, they are used to perceive light of high intensity, and also they are used to differentiate colors. So mostly during the day, we use cones for vision. So maybe if there is some darkness or maybe we are in some dimly lit room, so the cones cannot function. So the things now that function, they are the rods which function. So the function of the rods, they are used to perceive light of low intensity. That's why in the cat family, cats can be able to see at night. Cats have very many rods as compared to human beings. They have very many rods. That's why cat can be able to see in the dark or in dimly lit room. But we human beings, we have less rods, so it is difficult for us to see in dimly lit rooms. Let's get back now to the question now that we have understood what is color blindness. So the question, remember the question was asking, what is the probability that a first child will be colorblind? So as you can see the genetic cross, that is the genetic cross. So the probability that the first child will be colorblind will be 50%. If you can be able to see the offsprings in the genetic cross, you can see that the first offspring, XX, remember it's females, XY is males. So, the first offspring is X capital C, X small c. So that means this is a carrier female. The next one is X small c, X small c. This means now this female is colorblind. So for the female to be colorblind, all X chromosome must be infected. So, like for the first scenario, X capital C, X small c, it means this female is normal. Physically, they are normal. In fact, they are not even colorblind. That is the first scenario. They are not colorblind. But in their genes, they have the gene for getting an offspring who is colorblind. So maybe right now, if they get an offspring right now, chances are very high that the offspring they're going to obtain will be colorblind. That is what it means by someone being a carrier. So in the second scenario, we have X small c, X small c. So for a female to be colorblind, all X chromosomes must be infected, like in the second scenario. If all X chromosomes are infected, now that female also is affected, has the color blindness. The third scenario, we see that we have X capital C and Y. Now this is a normal male, because males can never be carriers. Why? Because they have only one X chromosome. It's either they have the disorder or it's either they don't have. So remember, capital C represented a normal individual. So now since this one has X capital C and Y, it means that this male is a normal male. The fourth scenario, we see that we have X small c and Y. Now this is a colorblind male. Now you see, it's either male has the disorder or doesn't have. Now this is a colorblind male. So the question was asking, what is the probability that a first child will be colorblind? So the probability that the child will be colorblind, remember these are chances, 1, 2, 3, 4. These are chances. It doesn't mean that the first child will be a carrier, a female, the second child will be colorblind, the third child will be will be normal, the fourth child will be a male. No, it doesn't mean like that. These are chances in terms of mathematics. There is 25% chance they're going to get a carrier female. There is 25%. Now, since we have now the female and the male who is affected, now there is 50% chance that they are going to obtain an offspring who is colorblind. The third scenario means there is 25% chance. So these things are 25, 25, 25, 25. It means chances. It doesn't mean the first offspring will be this, the second. No, it is just chance. So that's why we are being asked, what is the probability that a first child will be colorblind? So the probability the first child will be colorblind, the probability is 50%. Why 50%? Because the second offspring we have seen is a colorblind female. You only 25%. Now the fourth offspring you have also seen that is a colorblind male. That is also 25%. So if you add them, you get 50% chance that the first child they are going to obtain, that is the F1 generation, is going to be colorblind. So question B is asking, if the man's mother was colorblind, now the mother of now the man, if the man's mother was colorblind, what was her genotype? So this is simple. So if the man's mother was colorblind, so it means that the genotype of the mom will be X capital C and X small c. The mom was a carrier. So if the man's mother was colorblind, it will mean that the mother was colorblind herself. Remember, a mother is a female. So if a mother is a female, so remember, for females, they can only be colorblind if all the X chromosomes are affected. They can be carriers if only one X chromosome is affected. But now in this case, we are being told if the man's mother was colorblind, a mother is a female. So for females to be colorblind, all X must be affected. That's why the answer to this will be X small c and X small c, to mean that all chromosomes are affected. So again, don't you forget that females can only be carriers if one of the chromosomes is normal, one is affected. Females can have the condition if both X chromosomes are affected. Males can never be carriers because males have only one X chromosome. So it is either that chromosome is affected or that chromosome is normal. So always remember that. So the next question is asking that state the proportion, what proportion of all children from this couple is expected to be normal? So what proportion of all the children from this couple is expected to be normal? So the answer is only 25% of the children from this couple who will be expected to be normal.
Biology.